Thank you for that kind introduction. Thank you, Britt and Chris, for inviting me. Delighted to be here. So it's been about 20 years since the advent of functional brain imaging and the non-invasive studies of the human brain. And over the years, we cognitive neuroscientists have developed a number of different paradigms, attention, memory, language functions, and a whole slew of them. But underlying them are certain cognitive control processes that you have to bring to bear on almost every single task. The ability to attend to a stimulus, to engage with it, and turn on frontoparietal systems that are involved in maintaining information memory, maintaining rules, and switching in goal-directed, in ways that are appropriate to goals. And so this has been kind of the focus of some of my work in terms of more recently synthesizing findings across a disparate array of fields, and that's what I'm going to be talking about. At the outset, I want to acknowledge the key collaborators and funding, and I'll mention the individual contributions as I go along. And so what we are concerned with here is how can we understand the overall functional architecture of the human brain, its intrinsic spatiotemporal organization with the tools and techniques that we have, limited as they may be in terms of both spatial and temporal resolution. How do we study task-induced dynamics in the context of these underlying intrinsic systems? And what are those functional circuits and how do they get modulated by context-dependent modulation? And within this framework, we've tried to think about general models that transcend individual task domains to understand how the brain can flexibly process and allocate mental resources to permit dynamic selection of stimuli, thoughts, and actions in response to context-dependent goals and intentions. The notion that there are core underlying cognitive control systems that you have to bring to bear regardless of the domain, the task domain that you're confronted with. And so that can serve as an underlying idea for thinking about functional brain organization and how some of those core systems might relate to human cognitive function. And the particular kind of task domains I'm going to focus on as exemplars for more refined investigations of these systems are your standard attentional oddball tasks, both in the visual and auditory modality, your canonical inhibitory control tasks such as the go-no-go and the stop signal, the set-shifting stroop and working memory at a high level. As you add the complexity of these task domains, it really becomes much more challenging. But these point stimuli like the oddball and the go-no-go and the stop signal really give you a much better handle on individual trial types without getting into the the complexity of the temporal time course of processing, such as you might have in particular with the in-back task, for example, where you have to continuously monitor and update information in memory. But the idea is that some of these core control systems should operate in a very similar way, although you don't have access to the full dynamics, certainly not with fMRI. And then if this kind of framework or a model or the systems that you've identified have a role in cognitive control, they should, almost by definition, have an import into the investigations of psychiatric disorders because deficits of cognitive control and cognitive inflexibility are the core of many, if not all, psychiatric disorders. And so this provides an inroad into thinking about both cognitive function and dysfunction in a with a common underlying framework. If we can understand the functional organization that underlies cognitive control in these kinds of tasks, and we can show that there are deficits in the functional organization of these systems, then we have a common underlying model for thinking about cognitive function and dysfunction. And so this is an attempt to synthesize findings across studies over the last 2 decades in ways that differ from individual studies which observe different kinds of effects, particularly when you get to the whole brain level. There are a lot of different things that go on. And the question is, how do we separate the wheat from the chaff? And so those are some of the goals we set ourselves to a few years back. And the question is, how can we unify these themes of research to come up with common models, as I mentioned. So this is a view of the human brain, and this is recordings from Rakel's lab, but you could record it anywhere and display this. So this is with a temporal time course of 2 seconds, but now we have faster resolution that goes up to less than half a second. And you can capture this essentially gives you a notion of how dynamic the system is from the occipital cortex, the occipital pole to the frontal pole. And this kind of giant expansion of the prefrontal cortex that you have to really consider as we describe the architecture of information processing, particularly in relation to cognitive control. And so the notion is that the underlying dynamic intrinsic activity of the brain has some underlying structure. And it is on top of the structure that the cognitive control and indeed all information processing is built, right? And so, and we can actually simulate this, and we can model various components of it, not to full detail and exquisiteness that kind of defies our computational resource, not even to mention the kind of lack of information of particular parameters, which should underlie these models, but we don't have access to them at the level of individual neurons and synapses across this system. We can count the number of neurons and maybe get a better estimate of 84 billion neurons, but to get at all the parameters related to signaling at the individual neuronal and synaptic level is certainly outside the scope of maybe the next 50 or 100 years, perhaps. And so the first notion that these systems have an underlying architecture that's different from random comes from an analysis like a PCA or an ICA that you can do to decompose the intrinsic temporal course from each voxel. And in general, you see patterns like these. This captures the dorsolateral prefrontal cortex and the posterior parietal cortex, a system that we call this central executive network, and the frontal parietal network, more appropriately, the motor system, the frontal eye field and the parietal eye field, the default mode network, the visual system, and so on. You can tessellate the whole brain this way through this functional architecture. But, and there are various ways of using this technique to now map out your circuit and then say, this is the circuit I'm really interested in for this task domain. I'm going to try and map out the task-related dynamics, which might involve dynamics within the network, but also across the network. So these are statically defined networks. But in fact, both intrinsically and across tasks, you see a wide range of dynamics that involves the flow of information into each of these networks, the flow within the network and across networks. And so that's kind of one aspect of framing that we've been thinking about in terms of how to understand information processing and flow at this large scale. So it's not these are not data where you can actually infer things at the level of even tens of thousands of neurons. This analysis and these type of models are focused on much larger global scales in order to understand the architecture at the global scale. And perhaps they have import into thinking about how individual regions might function within the context of the overall fingerprint of each region. I should also mention in the context of what we heard this morning that the notion here of the function of a given area or node is now dependent not just on the intrinsic local architecture, but the areas that it is most strongly connected to. And those two features inform what the function of an area might be. So we have focused, and the focus of this particular presentation is on 3 different systems. One which I alluded to as the central executive network or the frontal parietal attentional network, a working memory network that's engaged when you have any complex rule-based processing where you have to keep information in mind and access that over a period that is longer than the period over which the stimulus was presented on the screen. The second system has been less well studied. So these systems have been studied in macaques, and now increasingly, at least the parietal cortex in terms of rodent working memory, with very interesting optogenetic manipulations. But this is a different system that's anchored in the anterior cingulate cortex and the anterior insula with subcortical nodes, which I'll get to in a minute, that we've called the salience network, and for reasons that I'll get to in a few minutes. The third system is also widely engaged or disengaged during cognitive tasks, and that's the default mode network that's anchored in the posterior cingulate cortex and the ventromedial prefrontal cortex, with nodes in the angular gyrus on the parietal side. Now these are all areas which have undergone quite a lot of expansion from even the macaques, and the angular gyrus in particular being a region that is part of this system, which, although you can actually identify the links between the posterior cingulate cortex, the ventromedial prefrontal cortex, and the macaques, some of these key parietal links are actually missing. And those are regions that lie actually at the interface of self-referential activity with language-related semantic processing. But that's not going to be the focus of this presentation. But this system is important in the sense that it's involved in internal mental thoughts, a process that you have to disengage when you do cognitive tasks. And so its importance here for this presentation is within that context. And so in most, for example, working memory or attentionally demanding tasks, these regions are actually suppressed, whereas these areas, the activations are enhanced. And we know that there are parallels also from electrophysiological studies. We published a report a few years back in PNAS showing that some of these profiles actually appear in electrophysiological ECOG recordings as well. So it's not an artifact of fMRI. That's the most important thing. So if you were to take a tool like Neurosynth, which allows you to do a meta-analysis of studies, and you enter the search term cognitive control, there's like 600 studies here, you see these core areas that I just highlighted before you in the previous slide, which is the anterior insula, somewhat surprising from the point of view of cognitive control. And now the thing is that along with the anterior cingulate cortex, and then you see more dorsally the dorsolateral prefrontal cortex and the inferior frontal sulcus, the middle frontal gyrus, which we typically demarcate as dorsolateral prefrontal cortex analog. And these regions come up over and over again across tasks. And it actually struck me that a lot of different functions were being assigned to these regions. So for example, in a task involving affective stimuli, the claim would be the insula is differentially responsive to processing negatively valent stimuli, and tasks involving pain would be things that it's actually processing pain. And when I looked at the literature, I felt that there was a much broader theme, and it actually appears across a broad range of tasks, although cognitive processes and affective processes interact often, but they also show up in very simple tasks like the oddball and the go, no go, where you wouldn't necessarily assign an affective value to any stimulus. So someone did actually a meta-analysis of quasi-meta-analysis for all the activated and deactivated brain regions. And here's a map kind of showing, what I want to highlight is the anterior insula, which showed up in surprising ways that the literature hadn't really focused on. And as I mentioned, this is kind of just another piece of evidence that this is, the so-called salience network, which is anchored in the anterior insula and the anterior cingulate cortex, and these more dorsolateral prefrontal cortex and parietal cortex are involved in a wide range of tasks. And so the mapping that I showed you earlier with the intrinsic functional networks now allows you to study this in a more principled way, because you can take an individual's data set and you can say, well, this is the architecture of these networks in this particular individual, and then you can ask how is that architecture constraining information processing. And so, and you can do that independent of task domains, so that across tasks you can build up profiles about what kind of task processes engage these areas and engage cross-network regions in a principal way. So this is a study that actually showed that, so when you have participants do these tasks, like an attentionally demanding task, like a stop signal or a n-back working memory task, you see activation in a number of different brain areas, including, so it's coactivation of the anterior insula, the dorsal cingulate cortex, and the dorsolateral prefrontal cortex and posterior parietal cortex. But looking at this from the point of view of intrinsic dynamics allowed us to separate these networks into subsystems, right? And so shown here in red are the anterior insula and the anterior cingulate cortex in one network, and then the dorsolateral prefrontal cortex and the ventrolateral prefrontal cortex, along with the posterior parietal cortex, here mainly the supramarginal gyrus as a different system. So although they are co-engaged during a task, intrinsically they're actually quite distinct. And the idea is that during task context, they start to interact with each other. The underlying reason for separation of these networks is really still not understood. I think it'll be very difficult for fMRI to disentangle that. And I think the availability of intracranial recordings, which is now happening through shared resources, is likely to be the way to push this forward. The other thing I want to highlight in terms of this so-called salience network, it is two important subcortical anchorings. One is the ventral tegmental area, and the other is the amygdala, two subcortical nodes that are involved in processing biological value and valence, particularly for negatively valent stimuli, and reward-based signaling. So these areas allow preferential, the fact that they're actually connected within a circuit allows preferential access of the anterior insula to the subcortical nodes that help to signal salient stimuli in the environment. And that is another reason why we term this system the salience network. The other reason was that, of course, it was activated during a wide range of affective tasks, where it was, as well as reward-related tasks, including monetary reward, social reward. On the other side, you have pain, negatively valent stimuli, and a whole range of other stimuli that signal biological valence, including thirst. So we noticed that the insula and the dorsal anterior cingulate cortex form a coherent network intrinsically. They're activated across a wide range of tasks, both cognitive and affective. And surely it couldn't be the case that this was a region that was involved in processing disgust or empathy or pain alone. And so we asked, can a system that's involved in detecting, that's sensitive to biologically salient stimuli also play a role in cognitive affective control? And through a series of studies, I'll show you that surprisingly, yes. And evolutionarily, this is noteworthy, because why would you want to have a control system or a triggering system that's not based on signaling of biologically salient value and valence and reward? And those are the stimuli that you want to grab into your attentional system and then facilitate access to higher order cognitive processing resources, whether they be language semantic based, which we've not studied sufficiently, I think, in the field, but more importantly, and one that's easily graspable and investigatable, I guess, is this frontoparietal network anchored in the dorsolateral prefrontal cortex and the supramarginal gyrus, which have been the mainstay of most of the studies of working memory and higher order attention in both humans and macaques. And so the theoretical idea here is that what this network does is it detects and orients attention to goal-relevant events in a flexible manner. And it is anchored in the anterior insula and the anterior cingulate cortex. It plays a crucial role in detection of salient events so that it can facilitate access to resources. And one of the interesting points about this system also, the architecture of this system is that through the links between the anterior insula, which is more sensitive to the stimulus contingencies, and the dorsal anterior cingulate, which is actually more sensitive to motoric and efference contingencies, you have a system that can play a tight link between stimulus detection and response processing. In fact, the anterior cingulate cortex is sensitive to reward to motoric manipulations, different types of responses in ways that the anterior insula is not. And so there's an internal dissociation here, which is quite difficult to disentangle unless you design tasks appropriately that manipulate the environment in one of two of these domains. So a very interesting example is that if you stimulate the dorsal anterior cingulate cortex, you get this, volitional sense of moving and wanting to make a response independent of any external stimulus. And that's been shown through intracranial stimulation studies. So this led us, this body of work that we reviewed and this notion of separation of circuits led us to the notion of a triple network model, which we think plays an important role in a wide range of cognitive tasks. And the idea is that the anterior insula and the salience network in general are sensitive to sensory and limbic inputs. Now, why certain stimuli have preferential access and engages the system is still not clear. How a salient visual stimulus or auditory stimulus may gain access to the system and enhance its response really needs deeper computational modeling at a level that we haven't really conducted. But the idea at the global level is that it then engages the central executive network to different extents depending on the task contingencies and load. And also actually, in a more natural environment, the subjective effort and load that difficulty that an individual experiences. So you may engage it more or less depending on how proficient you are at doing a particular task. And then the suppression of the default mode network in most task contexts. So these two systems are easily manipulable with external stimuli, and this system gets suppressed. The readout of this, the default mode network, is much more difficult because it actually encodes internally relevant information. And those are aspects that I won't focus on today. But together, they do play an important role in how we attend to stimuli. And so if you are over-engaged in the default mode network, you may not be able to switch attention to external stimuli because your mind is focused on something else, thinking about breakfast today and so on, for example. So, some of the ideas that we've put forward in the context of this model are there's a bottom-up saliency detection. We don't know exactly what the mechanisms and filters are that allow it, but certainly this region is sensitive to both the anterior insula and the dorsal anterior cingulate cortex are sensitive to this. And there's a switching of large-scale networks to facilitate access to resources, access to the motor system via coupling to the anterior cingulate cortex, and the anterior cingulate cortex that links to other motor control hubs, including the mid-cingulate cortex. And then the other aspect is interaction of the anterior insula with the posterior insula, which is more tightly linked to the hypothalamus and the physiological reactivity to salient stimuli, which we think, and there's some evidence, that it gives rise to the sensation of things that have happened in awareness, of physiological reactivity to salient stimuli. And that's work of Bud Craig and others. And I think we'll be hearing more of that as many of those processes are really clearly relevant to psychiatric disorders, if not the interface of cognition and emotion. So one of the things that we did with this data, now that we understood the overall, we have a sense that this architecture of these large-scale systems is highly reproducible. You can take any individual's brain, including patients with psychiatric disorders, and at least to the zeroth order visual extent, you'll see very similar patterns of response. It's a question of how these systems are connected and how they interact with other systems that gives rise to cognition and also underlies pathology. So in a very simple visual oddball task, we examine causal influences from the insular cortex to other regions. So we took two nodes, two canonical nodes from each network. So here's the salience network nodes, and these are the default mode network nodes in yellow, and these two nodes from the lateral frontoparietal cortex, frontoparietal network. And what we saw was that there was a very strong causal drive. This is done through Granger causal analysis, but we have other methods that have really tried to tackle this much more rigorously mathematically. You see this kind of very strong causal drive from the anterior insula. So this gave us the first hint that the system is actually capturing something related to attention and then triggering other systems. Now, this actually makes it evident across these two tasks, the one that I just showed you, as well as an auditory event segmentation, where there's a brief pause between episodes of auditory sequences. And you can see that in both cases, so this is not really modality specific, this is really counting the number of out degree flows in the graph, in the directed graph versus the in degree. And you see that we have a very strong causal drive from the anterior insula. So it's a causal signaling mechanism, we think. And obviously more data from electrophysiological studies, intracranial studies is needed to verify all of this, because that's really the ground truth. But what we have done is to actually push the envelope with trying to get data outside of things in our lab. And so to push the boundary with different types of tasks, we used a open fMRI source that curates maybe about 100 data sets now that labs around the world have made available. And we use 3 that are really canonical for cognitive control: the stop signal task, which I'll describe in a minute, and the flanker task. And we said, are they common? If this is the general idea, do these same processes come up across these 3 tasks? And so here's kind of, you're probably all familiar with this, but the stop signal task, it's like a go, no go, but after you receive the go signal, you're given either an auditory or visual cue to stop a response, and you can assess the time a subject requires to withhold response, which is called the stop signal time. You can assess it using a race model. And here's just kind of the extended design, the 2 different types of stop signal task, and a flanker task where you have congruent and incongruent conditions, and you have to make response in the direction of the central arrow. And obviously this incongruent condition is more difficult. It's another canonical cognitive control task. And for our purposes here, what's really important to note is that if we look at the brain responses to these 3 tasks, and you look at a logical AND, you see this common pattern of response, fairly right-sided on the anterior insula and the anterior cingulate cortex extending to the dorsomedial prefrontal cortex and the frontoparietal system. And so you can see this pattern consistently across all the tasks. And although there's variation across tasks, these are very strong common features. Now, we've developed some new models to go beyond Granger causal analysis, because Granger has several weaknesses, including the fact that you can't assess trial-specific response causal influences. And second, when you look at causal influence in fMRI data, you do have to control for regional variation and hemodynamic response function. So if an area has a sluggish hemodynamic response function, kind of time to peak of the signal, could be slower, although it was initially, it was activated earlier than a region that had a much faster hemodynamic response. This is just a function that couples the underlying neuronal activity to the fMRI signal. So you really have to be mindful that what you're observing causally is with the Bowl fMRI signal, not the neuronal signal. But so how can we push this? And so we developed a model where this is kind of building on other work in the field, where we have input stimuli, you have 2 populations that are responding, you have underlying latent states. They're not quite neuronal signals, but they call it quasi-neuronal signals. and then you want to control for variations in HRF. And we did that through a state space model. I won't go through all the details, but I just want to tell you that we've gone through great extent to try and overcome limitations of fMRI in terms of response, and by looking at convergence across tasks and using multiple analytic tools. Essentially what it is that you have a latent signal where you want to assess the coupling of the response. So CJ is basically the causal influence of one region over another. But you're assessing in some unknown latent space when you have the observed space Y, and the coupling between them is through this hemodynamic response. And we've validated this using a model like the Wilson-Cohen model. We took two populations of neurons, and then you have some underlying dynamics with one leading the other, and then you want to convolve that with the HRF and variations in HRF, and you want to downsample it, and then you want to ask, is the causal influence that you observe in this neuronal model reproduced in the fMRI space? And that's essentially what we try to do here. And with these computational models, we were able to show that the method works. And then the other thing we did recently was to use optogenetic fMRI with whole brain fMRI recordings and stimulation of the motor cortex and recording in the motor thalamus. And you see these three rodent examples showing that we can actually recover causal influences. And it's just kind of proof of concept that the method works. And so we use this technique, it's not to belabor the technique, but to illustrate that we can actually push the envelope of temporal resolution through having different tools that acquire, control for potential confounds in data. And again, we see this pattern, which is, I think, easier to look at here in the bottom graph. Very strong causal influences, again, from the right anterior insula across these three task domains. And if we look at, now we can actually look at task-specific differences across the task conditions. And we see here for the more difficult stop versus the go trials, you see this across all these pairs of regions. The most consistent effect is signaling within the salience network from the anterior insula to the anterior cingulate cortex. And furthermore, this is behaviorally relevant because the strength of this causal influence also predicts behavior response, in the sense that the stronger the signaling effect, the more it's reflective of task difficulty, which then maps onto the subsignal reaction time and the congruency effect, which index task difficulty. So the summary of this part of the study is that the right anterior insula in particular acts as a causal signaling hub to the extended frontal control system, mainly the frontal parietal network. It's also sensitive to the load conditions. It's related to behavior. And we think that these are some pieces of evidence that suggest that the system plays an important role in implementing cognitive control. And we have other studies. We've actually pushed this with other types of tasks, including disentangling proactive and reactive control. Proactive when you have a task contingency that builds up. So if you receive 4 go trials, you're likely to expect a stop signal to occur in the subsequent trial. So you can use a Bayesian model to analyze the probability of, you can do this at an individual subject level, probability of a stop signal occurring. And that gives rise to a proactive signal where the signaling direction is actually reversed into the anterior insula from the ventrolateral prefrontal cortex. Whereas on a reactive task, which is most of your go trials, you see this a stronger influence from the anterior insula. So even within neighboring regions, you can dissociate effects related to different aspects of control. So now you may ask, this is all good. This is fMRI data. We don't have the temporal resolution to be sure that there's a network signature of the salience or any other network. And so very recently, we've been lucky to get 2 open source cohorts independently acquired at the Montreal Neurological Institute. and the UPenn Memory Consortium. And what we did, we were lucky that these areas from our canonical, the three networks, these canonical regions were actually fairly well represented. As we imposed this criteria, they had to be at least 5 individuals who had data from a pair of regions. And then we subjected it to a whole set of analyses. And so mainly they relate to looking at the correlation within and across networks using linear and nonlinear measures, the phase locking value on the nonlinear side. But also looking at the temporal dynamics using complexity measures and mutual information. And then, interestingly, a pattern that's called joint recurrence rate. So how likely is a pattern that occurred at one time to occur later? And so you'd predict that within the network, you should have stronger occurrence rates of these patterns. And so we use these measures to examine the integrity of this model, right? So focusing on the salience network. And so what we found here was that, so this is looking at correlation, this is the MNI cohort, this is the UPenn cohort, and this is looking at, so we focused on the delta band, given the low frequency nature of the fluctuations in fMRI. The studies have been highly variable about the frequency of the underlying representation. There's a lot of studies on human scalp data. None of them are really compelling or believable. And there's also the notion that there might be, fluctuations in gamma band and so on. But the strongest effects we found was in the delta band, 0.5 to 4 hertz. And this just shows you the replication of the data, the AI and ACC, much stronger effects. The correlations compared to the intra-network correlations compared to the inter-network correlations. This gives a sense of the network integrity. That's what you'd expect. And we can look at a phase locking value computed through an instantaneous phase metric, the Hilbert transform, and you see the same kind of pattern. And then we looked at the recurrence rate, which is the pattern that occurs in one region. Does it recur in another region with greater rates? And yes, that's indeed the case, that the AI and ACC have much stronger internal recurrence rates of patterns. And then we looked at a causal metric here, in this case the phase transfer entropy, which also showed that the AI has very strong, and the ACC have very strong causal influences on other nodes. The other thing that we did was to look at how fast is the signal decorrelated in an individual region. And you can see that the decorrelation time is actually lower, so the underlying dynamics is actually faster. And we have both a nonlinear and linear measure of it. And then we have the sample entropy, which is a measure of the complexity, which is also higher. So there's a faster dynamics that's occurring in this region. We have some task data from the UPenn. We're going to analyze that at some point to look for how these features change with essentially episodic memory tasks. But the notion is that we have now a signature, a electrophysiological signature of the spatial-temporal integrity of the salience network, and it recapitulates several of the features that we see with fMRI data, and which in some sense there had to be electrophysiological signature because of the ubiquitous and highly reproducible nature of these networks observed with fMRI. They're not ephemeral. You can actually record them every single time. And so there must be some underlying electrophysiology. And this is one hint of a process, and there's probably many more to be discovered as these data become more widely available. Very, very difficult to get data. And so so the next thing we did was to look at the whole architecture of the functional organization. So far we've been mainly focused on the static network organization. And now we asked what's the nature of temporal evolution of interactions between these networks. And so here what we did was to identify about two 164 regions. These are based on a set of areas that are involved in cognitive control and visual auditory processing from a WashU group. They are known as the Power ROIs, if you're familiar with the domain. And so we extract time series. We do a windowing. We do a community detection. And then we ask how likely are two nodes to be within the same community over time. And then we can then derive measures of temporal flexibility. We can also derive measures of spatial diversity. How widely is it interacting with other systems? So both in time and in space, we can assess the level of flexibility. I'm happy to discuss the details later. And so what we saw here was that two sets of nodes that showed very high levels of flexibility. So the power ROIs are actually mapped into 12 canonical networks across the brain. Some of them I've alluded to before, the salience network and the dorsal attention network, which is essentially the frontoparietal network. But the two things that really stood out was the salience network nodes. There are about 4 or 5 nodes in the regions of interest in the anterior insula and about 3 or 4 in the dorsal anterior cingulate cortex. If you pull them all together, you derive a network-specific flexibility measure. And the two subsystems that showed the highest level of flexibility were in the salience network and subcortical regions, including the caudate and putamen amalgamated here. And we haven't fully unpacked those. And this is replicated in a second set of data from the same individuals. And there's kind of an interesting feature that appears here where these regions stand out in terms of their temporal flexibility and spatiotemporal diversity. And this is kind of illustrated here in terms of the specific nodes that I've been talking about. Moreover, the flexibility of these systems, but not the subcortical nodes, was really related to cognitive flexibility as assessed using these NIH toolbox measures, processing speed and card sorting and flanker in a canonical correlation analysis. And this was also replicated in the second session. So the general idea is that this is individual variation in flexibility of these systems' predictive cognitive flexibility. Another handle that this system is really important for not just cognitive task performance, but in general cognitive flexibility. And the idea is that in relation to psychopathology is that deficits in the dynamics of the system and how it interacts with other brain areas may contribute to psychopathology, which is kind of the next part of the talk that I want to focus on. So the idea that you can actually assess using these tools dynamic flexibility, and the salience network has a relatively high level of flexibility compared to other systems. It also interacts and samples other brain systems more widely. The notion that you want to sample events occurring in different parts of the brain. This is still a hypothesis. So a system that has a high level of flexibility in this sense should be able to perform a more integrative function in terms of detection of salient events and then orienting attention to them. So with these ideas and background in mind, I want to turn to the notion of what happens to these systems in various psychopathologies. So most of this work is still with intrinsic functional imaging data and a couple of studies that we're working on are looking at task data and how these intrinsic flexibility is related to task-related dynamics and flexibility. And that's still part of our ongoing work. But the first ways was to see if there's a correlate of psychopathology in these in the intrinsics integrity as well as the dynamic interaction of these networks. And so this is just kind of to refresh you this kind of model of inputs into these systems, and we've shown that there's a very strong signaling outflow into the central executives network and suppression of the default mode network. And based on this, we've proposed what I think might be a very general model one of the more general models of thinking about major psychopathology. And the idea is very simple. So weak mapping of salient stimuli fails to engage or over-engages the salience network, which then causes signaling activation of the frontoparietal system so that you then over-engage it. Or if it's weak, you don't suppress the DMN and come out of your internal mode of thinking. And all of these processes through both external and internal valence stimuli can impact how these other systems talk to each other and integrate information. So that's kind of the general theoretical framework, and over the last few years we've been trying to unpack aspects of this in various disorders, and I want to show you some examples of that. So this is the first study focusing on children with autism, and what you can readily see is that these networks are actually identified in children with autism. It's not that they don't have these systems, or that's not the level of dysfunction. The level of dysfunction is the integrity of these systems and how they interact with other systems. So one of the things that actually struck us, which actually has, I think, changed the field in some ways, is most of the mouse models of autism have been based on imbalance between excitation and inhibition, the Rubinstein-Merznick model, which gives rise to hyperactive function in a given area. But the tools in the rodent were not sufficiently developed at that time, 2003, to kind of push this into the level of the global signaling. So if you have a hyperactive region and it signals to other regions, you might see hyperconnectedness with other brain regions, which is a metric that we can actually assay here. We can also obviously assess hyperactive fluctuations in a given area. But the compelling results in the field at that point were, there's a lot of hypoconnectivity, underconnectivity, and so the information is not processed or getting access to the system and so on. But we actually found the reverse. A lot of, like you would expect from these EI imbalance models from the mouse models of autism, we actually saw hyperconnectivity in a number of different systems. And when we looked at which system actually is the most predictive in a cross-validation sense of the disorder, it was the salience network that actually stood out the most. And moreover, the degree of aberration in the system is actually also predictive of restricted and repetitive behaviors. And this is a component that I'll try to unpack. So there are two components of restricted and repetitive behaviors in autism. One is motoric, the hand flapping. But there's also a cognitive aspect, which is circumscribed interest, restricted interest focused on one particular thing. It might be calculation, or it might be drawing, or it's just hyper-focused on one thing to the exclusion of everything else. And so this was the symptom, the clinical phenotypic feature that was captured by this network. Now, the other thing in relation to this is one of the first studies of this sort where we asked, if this system is hyperconnected, then maybe when you ask the participants to engage with a socially relevant task, in this case detecting an oddball face stimuli, the modulation of these circuits might be impaired in children with autism, because it's hyperconnected and it's not able to modulate the response. And that's kind of what we found here, the degree to which they are unable to modulate their response over the hyperactive connectivity state, the stronger their clinical symptoms. And then now we've gone on to unpack this in using dynamic functional network measures in children with autism, children with ADHD, as well as schizophrenia. They all focus on these three core networks that I've been talking about, but they manifest themselves in different ways that we're only beginning to understand now. And in ADHD, we actually, building on our work on cognitive control, we have several studies looking at the stop signal task and looking to see how the underlying dynamics might influence how children with ADHD might process that information. So I don't have time to unpack all of that today, but I'm just going to give you a glimpse of the findings from the three disorders to convince you that there are fairly strong signatures that are representative of the clinical phenotype in each disorder. And although we don't understand why they lead to such different roads in terms of the clinical manifestations, there are common underlying themes in terms of the systems that are impaired. And the goal is going to be, I think the next several decades, I think, going to be to try and unpack why this happens. But at the core of it, of course, is where, what I mentioned when I started out, that there is this dysfunction of core cognitive control systems, and that's leading to downstream impairments. And at the core of it is the salience network, which we've tried to unpack in many different ways. So I'm just going to wade through this relatively quickly in the interest of time, but that was the kind of the main result that I wanted to convey. So in this study, what we did was to try and unpack the role of the frontal control circuits that I mentioned and a motor circuit, and trying to unpack the two aspects of the clinical phenotype that I mentioned, the motoric aspects and the cognitive aspects related to restricted interest. And so here's one of the kind of tools that we've been using in these kinds of studies. You take the time series data from these systems, and then from each system, their canonical nodes as a way of studying the interaction of these systems. And then we can either use a window-based approach or a hidden Markov model, which gets you the latent states. And from those latent states, you can get the community structure of each state. And then you can, through a thresholding or other approaches, you can get at the overall community structure. And so one of the things we see here is this kind of, what we saw in the static state, which is that these networks are completely dissociated from each other. There's no crosstalk between them. And then there's the other state, so we call this the segregated state, right, that we started out with earlier. And there's the integrated state where these nodes are talking to each other. So this is how we've reduced the space from this complex set of states into two canonical states. And then we can assay, what's the underlying circuitry, what's the covariance structure, what is the transition probability, and so on, and how is it related to clinical symptoms. So these are subjects, and this is time, and this is showing kind of the occurrence of the segregated and integrated states over time. And I just want to refer you to this part of the graph showing that there are significant differences between the, let's just look at the occupancy rate of the state. So this is something that the HMM model actually gives you. It gives you the occupancy rate and the transition matrix. You can see that the children with autism tend to stay in the segregated state, whereas the typically developing children, TD children, stay in the integrated state. So this is the segregated state, and any of these could constitute the integrated state. So there's differences in the underlying states. And this is just kind of shown here that the children with autism tend to stay in the segregated state where the networks are not talking to each other. It's kind of a model for thinking about how hyperactive circuits might really be dysfunctional. It's just hard to engage and have crosstalk between these systems. That's the general idea. And that's also what we hinted at when we looked at the social oddball task. And now we can actually look at the transition probabilities. And one of the things that really strikes you is that children with autism, when they're in the integrated state, they have higher transition rates into the segregated state. And the reverse is true in children with typically developing children. So this is a transition matrix that underlies the phenomena I just described, which is that children with autism tend to stay, because they have hyper-connected circuits, they tend to remain segregated. And it's just much more difficult to engage crosstalk between them. And just very briefly, we've looked at this in the motor circuit as well. There's a different characteristic. But we have an interesting behavioral double dissociation here, in the sense that the aberrant dynamics of the cognitive control circuit with the salience network nodes and the frontal parietal nodes predicts the cognitive phenotypic features, but not the motoric. And the motor circuit, which involves the SMA, the motor cortex, and the cerebellum, and the thalamus, predicts motoric deficits, but not the repetitive behaviors in the motoric domain, but not the cognitive domain. So these are hints that there are different systems that, it's again going back. So those network nodes are all derived based on static network organization. And we're looking at the dynamics and say there are dynamical features that are predictive of clinical phenotypic features and can also be dissociated. So the general idea here is that you have an intrinsic hyperconnected system, poor task modulation, poor switching between states. And could this be an interesting way of thinking about childhood autism, certainly? And I think it has the potential to now link back to the rodent studies, which people are now doing with different types of manipulation, genetic manipulations, and whole brain recordings. So there's a possibility of bridging this translationally as well. So we've now gone on to extend this work to look for features in these three networks in ADHD. And it's a study that we published earlier last year. And again, these networks can be identified. This is a primary cohort, replication cohort. You can see there's a very strong correspondence between them. This is, if I were to put you in the schema, I'd see very similar things. But if you look at the underlying connectivity and dynamics, you would start to see important differences. So this is in a, so here what we did was to look at the interactions a little bit more specifically using a window-based approach. And so what this does is it's looking at the extent to which the salience network is coupled to the frontoparietal network and decoupled from the default mode network as a metric of network interactions. And what we see is that the children with ADHD have lower network interactions, and that is actually predictive of these inattention symptoms. But even more strikingly, if you look at the dynamical features, so here in this case using the window-based approach, you see that whereas typically developing children fluctuate between 2 or 3 states at the most, children with ADHD, their states are highly volatile. They fluctuate across many different states, and that's shown here. The mean lifetime of the states is much higher, and there are fewer states. That's actually spread out across multiple states in children with ADHD. And we've replicated this in 2 different cohorts. And now you can assay this network interaction metric in a dynamic way as well. You can look at the mean, and the variability. So the mean was higher in typically developing children, but the variability is higher in children with ADHD, right? So, and then if you put those things together, you see this, the variability is highly related to symptom severity. And so the notion that there are highly volatile states is a different feature from what we see from autism, highly volatile states. And although this is not the time scale of behavior, we don't know what the time scale of these type of states is and what exactly it's encoding. Perhaps it's some aspect of behavioral fluctuation that's not contingent on the 300 milliseconds you need to process a go stimulus and things like that. But there's an important behavioral signature of the aberrant dynamics here. And so this is just kind of a summary of that. But the key notion is that this widely distributed across states. And this is, finally, this is a study. This is built on the notion, Sitij Kapoor's notion of aberrant saliency in schizophrenia, the idea that you would assign abnormal valence to a stimulus that does not either exist or is generated internally through some hallucinatory mechanism. And so it's been a very prominent and interesting model. He's really focused on the ventral striatum. We looked at the cognitive control system, and I think the next phase is going to be to look at the interaction of the ventral striatum with this. And I'll show you a very interesting piece of data related to that just came out on which we can actually build. So do I have a few more minutes? Okay, so just very briefly, there is a signature of this, and we actually were able to replicate this in a second cohort. This is one of the interesting things about this intrinsic functional imaging data is that it's now all over the place. People are sharing it. And the first thing I think we want to do is to have a replicable finding before we, and we certainly cannot collect all kinds of data in our lab, and this has been a way to extend beyond this and to test our ideas and models. But there are some domains where we're actually applying this to the task, ADHD in particular, and autism as well. So so here also we see actually a very similar profile of high variability of states. So a state, by the way, is characterized by a particular covariance structure, much more low occurring mean lifetime states were spread out across multiple states, fewer states in typically developing children. And then you see this same profile, that variability in network interactions related to clinical features, in this case, the positive symptoms. And this is also replicated in a second cohort, which we got from an NIH-funded study. So, and then you can use these metrics to actually have a reasonable level of classification. It's not designed to be a biomarker, but just to illustrate that there's enough power to separate the groups. So this kind of, what I want to leave you with was this kind of model that we've come up with that has explanatory power across many different disorders. And some we've unpacked in a task context, but our first pass was to really understand the intrinsic architecture. And we're seeing dysfunction in the interaction, although the manifestations are different. These cognitive control systems and their dynamic interactions are impaired in the three disorders we've looked at. And there are other labs which are looking at other disorders. And surprisingly, this signal is there in virtually every. What drives it, what causes it, is a mystery. And finally, just like 3 slides here showing the generality. I mean, one of the reasons we developed this model, and it actually has, in some sense, cachets in relation to psychiatric disorders. And if you look at transdiagnostically the brain areas that are disrupted, so if you do a meta-analysis of several hundred studies that have been published across all disorders, you see that these are the same systems that show structural morphological deficits across disorders. And it's really quite striking. It's really the salience network, the DMN, and the lateral frontal-parietal cortex. And here's a more recent kind of incarnation of this that just came out. If you look at the altered connectivity of each of these these networks, if you look at the DMN, it's the connectivity with the salience network and the insula that's impaired. If you look at the frontal-parietal network, again, it's the insula and other frontal-parietal regions that are impaired. If you see the salience network, it's another node in the salience network and the dorsolateral prefrontal cortex. In fact, this is 8,000 patients. And so I think it really brought all these pieces together in an interesting way. And this final piece, which I found really very interesting, was a combined PET fMRI study where they showed, I'm just going to go through this quickly, saying that the salience network connectivity is related to mesolimbic dopamine function, which they index through raclopride. And so this goes back to the earlier slide that I showed with the salience network having the subcortical ventral tegmental area. Perhaps there are comments, we haven't really fully mapped that yet, in relation to processing reward and other dopamine-related saliency effects. So this kind of helps tie in some of the notion of saliency from yet another perspective. And we'll see more of this, particularly, I think, in the context of schizophrenia, certainly, but also ADHD, where there's a complex interaction of reward and control circuits. And so I just want to conclude. I apologize for going over. What we started out with was saying is that there's an underlying architecture of the human brain organization. It's not random. There's a method to the madness. There are particular functional systems. They come up over and over again in cognitive tasks, affective tasks. And I've highlighted the role of the salience network and the anterior insular and the anterior cingulate cortex in attentional capture and then triggering other systems that are involved in higher order control. And it's a flexible system, and its inflexibility is related to a number of psychopathologies.